case studies as well. So yeah, check out that review of the homework for week 3 document for some of those links, as well as our folders.
Yeah, there's some of in the beginning, there's like a strongly suggested homework, which has a couple of resources. And then there's a lot of optional homeworks, as well as a lot of optional readings and resources that you guys can look on. And there's just, like Mia said, a whole plethora of really amazing information from just the past couple decades, really. And so it's really wonderful to have all of that easily accessible.
So Cade mentioned that he saw the folder, the case studies folder that I mentioned, but then Ellen asked if the folders on Fire are live. Has anyone had trouble accessing those folders? Yeah, then they are live. They should be. Ellen.
I have a question about turning in homeworks. I saw that, let's see, I uploaded my map onto the week 2 one, but then there are a lot of questions too. So I ended up just emailing Brendan the answers to those, but is that what I was supposed to do?
Yeah, I think so. I think that's fine. With Brendan's homework, I believe, yeah, any questions or answers in that diagram was supposed to be sent for him for his own personal feedback. So yeah, great job.
Okay, thanks. Yeah, and it was funny because I found it really hard to map my food like system, I guess. I ended up making a really messy spread. And then what I finally decided on like answering the questions about wasn't about food at all. It was about fire. So I'm glad that we're getting to week 3 because I came up with, like, we have a lot of dead spruce trees in acreage from the spruce bark beetle and also from drought.
Yeah.
And so that's causing a big fire problem. So there's a lot of crews going through and clearing out the spruce trees and like chopping them down. But then those slash piles aren't decomposing very fast. Like last fall, and I saw slash piles that were 10 years old and still weren't breaking down. So I was thinking we could, if we inoculated those with mycelium, they could have better moisture retention and break down faster. But then I came up with the problem that I don't really know what grows very well on spruce trees. So if anyone has advice for species that grow well on spruce trees, I'm curious.
Awesome.
Yeah, there's been a good amount of research.
Sharing all of that. And
seems like we just froze. Are people here? Okay, I see a lot of movement, but Taylor seems frozen. I was just going to say that there are some studies about fungi that grow in spruce forests, mostly ectomycorrhizal fungi. However, there are a lot of decomposers out there as well. One of the papers that's in the fire folder currently by Sandra Holden, is looking at the effects of fire in I believe it's black spruce and white spruce forests, and she looked at ascomycetes and basidiomycetes that were found there. So that might be helpful, even if it's not specifically using spruce as a substrate for cultivation. Okay, great. So I feel like we should go ahead and get started.
So I want to include, it's 5:07, so we should get started, Mia?
Sounds great. All right, so I'm going to go ahead and share my screen. Once again, my name is Mia Maltz, and today I will cover fungi from a community ecology perspective. And I'll be relating it to environmental drivers of microbial diversity. So here I'm going to go ahead and share the screen. Let me see here. Everybody can see my screen. Let me move our faces into the corner so it doesn't block what I can see. And then we'll go ahead and launch right in. So I am a researcher. I'm a mycologist, and I work generally with fungal ecology. So when I started working on fungi, I was interested in the role of fungi in ecosystems and how sensitive fungi are to various types of disturbance. And then through understanding which fungi are sensitive to various factors or human activities that degrade landscapes, we could then target our restoration to produce habitat for these fungi. You know, certainly we know that we're losing biodiversity at an unprecedented scale. And in that, we have a real sense of the number of animals and plants that there are in the planet, and those that are being conserved. So fungi have been on the planet for a billion years, and there are over a million species of fungi, and 98% of them are undescribed. So as fungi are sensitive to disturbance and we're losing them, it's important for us to get a sense of who they are, what they're doing, so that we can conserve their habitat. Currently, there's only 50 species of fungi that are being considered for conservation status globally. But as we continue to do the types of research that I'm covering here in this talk today, hopefully we'll get more of a sense of what the factors are that structure fungal communities and ecosystems. So I would like to, I can't see myself or anyone, so I'm just gonna redo this again. Okay, there we go. That might be working. All right. So, and we know that these factors are changing landscapes, whether it's glaciers melting or fires that are coming through and affecting whole biological communities and inhabitants of some rural and some very remote regions. So today I'm going to talk about the type of research that I'm interested in, research with fungi. That's my red thread that moves through all of the work that I do. I'll talk a little bit about some of the basics of experimental design when you're working with fungi, but also just as a field ecologist or a lab scientist, what are some things that we may want to think about or consider? I'll cover functional traits of fungi and fungal ecology and how we may leverage fungal processes for restoring degraded landscapes. And then also fungi and post-fire ecosystems. And then we'll finish up by giving a little overview on molecular microbial ecology. And any one of these topics I could probably talk about for a lot longer. So I'm excited to cover these things, and then if some questions come up, we can talk about it and follow up in further sessions as well. So how did we get ourselves into this mess that we need to turn to the fungi as allies? Well, certainly our human activities, our insatiable need for natural resources like oil, mining, for minerals, and even development and construction has had a profound impact on the air we breathe, the water we drink, and the soil we grow our food in, and that all walks of life rely on for our survival. So as we link waste products with food, we think about fungi, especially fungal decomposers, as main agents for recycling organic material. And for me, when I think about what sort of food I might be comfortable eating, I think about, what did I grow up being accustomed to eating? And oftentimes my comfort food that historically I ate growing up, like for me, it was mac and cheese. The other day I was talking with someone who said it was rice. These different comfort foods, they give us nourishment and sustenance, and the fungi, what they've been historically exposed to, whether it's organic materials, wood waste, these materials can become their food source. And when they're historically exposed to something over time, they can get comfortable eating it and growing and proliferating, and that food will fuel their growth. So when it is an organic pollutant, that makes a lot of sense that fungi can view these organic pollutants as food sources, because it's very similar in the chemical structure to wood or woody debris or plant litter. Now, there's this concept in microbial ecology that's called microbial infallibility. And microbial infallibility is it's this basic tenet of microbial ecology that, well, first of all, that microbes are everywhere. So there isn't a habitat where we won't find microbes. And second, that there isn't an organic compound that's naturally occurring that can't be degraded by some sort of microorganism. So this concept of microbial infallibility, it really gives me a lot of hope that all naturally occurring compounds or molecules can be degraded by a microbe. Now, what about the environmental conditions? How do the environmental conditions affect how they can view these different compounds as food sources? Well, certainly when the conditions are really harsh, the growth rate of an organism might slow down. There's some sort of trade-off that's going on where the organism will need to slow down their growth and put a lot of energy into survival. So when the conditions are favorable, when all of their needs are met, then they can go out and forage for food that'll fuel their growth and proliferation. Now, in our ecosystem, there's naturally occurring substances, but there's also foreign substances that are permeating into our ecosystem. And these are substances like plastic and certain pharmaceuticals. These are xenobiotics that are artificial compounds where the bonds that are found in these compounds are not naturally occurring. So when a microbe might see an organic material or some naturally occurring molecule as a food source, it may not have the skills to be able to break down some of the more difficult to degrade artificial substances. However, if we can move from the field to the lab and work with microbes and be able to allow them to assimilate to grow on organic materials, we may also be able to get them to grow on plastics or other artificial compounds. So this is sort of the foundation of thinking about how we may leverage fungal processes that are naturally found in nature to be able to close some loops within the industrial ecology. So just because they can do it, if a fungus or a microbe can break down plastic, and this is, I don't know, I don't have a cursor here, but this little circle in this house really stands for a petri dish. Like if you can get it to work in a petri dish, can you translate that to working in the field at a small scale or a large scale, really scaling up? So that's where some of the greatest challenges in our field lie. Just if we find microbes that have the ability and we are able to characterize that and document that and show that it's possible, sometimes that ability doesn't directly translate into action. So I want you all to consider that when thinking about moving now from the lab to the field. And why we may want to mimic certain environmental conditions. If I find a microbe growing in an estuary where there's brackish water, it's very salty, that level of salinity that's in the soil at the bank may be something that we would want to mimic in our microcosm or mesocosm experiments, just creating these small scale little ecosystems in order to evaluate whether some process can take place. And certainly when moving from the lab to the field, there's a number of different ways that we could then try to promote the ability of the microbes to do this work into the action of either remediating a toxin or promoting some sort of ecological function. I wanted to briefly touch on this concept. So we just talked about how favorable environmental conditions are important and how when you move into an environment, the environment is going to dictate who grows, who proliferates, and who may perish. Here I show this group of organisms. These shapes just represent different species or so. And the square, the triangle, the star, and the circle may get filtered out of the environment because it's too acidic. And in that case, the square won't be able to pass through the filter. The pH of the environment filtered the square out of the community. Certainly, we were just talking about salinity. Salinity could also promote the growth of some microbes, but also deter the growth of others. And similarly, there's some microbes that have traits that they prefer to be cold. So these psychrophilic yeasts that I find sometimes in these very inhospitable alpine environments, psychrophilic, like loving cold, versus thermophilic organisms that love to live where it's hot. There's desert organisms, and then there's even extremophiles that live by thermal heat vents, like out in Yellowstone. So factors in the environment will filter out members of the community. Similarly, these interactions among different organisms in the community will also drive these patterns. So we may have the square and the star competing with each other, and then they figure out how to coexist. And the star may actually be a predator that's able to see the square and the triangle as prey. So, this is just simplification of these concepts of environmental filtering and biotic filtering. And a species, a mushroom, a fungus, a microbe must have a specific combination of traits in order to gain membership in the local community and to be able to persist given the environmental conditions at that site, and then the features or the characteristics of the biotic community. And that will enable them to survive in the community, given the favorable environmental conditions. And for us as scientists, as fungal ecologists, we have this opportunity to make these pairings, to characterize the environment and also characterize the traits of the microbes that are capable of surviving in these environments. So I spoke to a few of you just briefly when we were getting on the call about how I'm interested in developing a tool that would look at the features of a compound, a pollutant, or some sort of threat. It could be a pathogen, features of the environment, and also features of the microbe or the microbial community. So you can do some strain selection within the context of ecological restoration or microbial remediation. So I want you to think about that lens when you analyze some of the case studies that are in the folder. Think about the compound of concern, features of the environment, and then the traits of the microbes, because in developing this tool, it would be phenomenal to be able to do this in collaboration with you, people that are very interested in this field, and come up with a choose your own adventure story here, a dichotomous key activity for remediating degraded landscapes that's really focused on the characteristics of the pollutant, the environment, and the organism. Because certainly conserving sensitive habitat is challenging. And as we move forward now in the talk, I want to just orient us as to where we are here. We're right at the beginning. That was all sort of just some background in what motivates me, what motivated me, how did I get here, and now we can really launch into some of the meat and potatoes. Or tofu and kale, whatever you prefer. All right, so I'd really like to be able to see you guys or see the chat, so I'm just gonna move this around again and play it. Okay, that's a little bit better for me. Is everyone doing okay? Does anybody have any questions at this point? Good, okay, great. So biological communities differ from populations or individuals, and I consider myself a community ecologist. And communities are assemblages of species, multiple populations that are living together. And maybe a rotting log has a lot of fungi. We might see the fruit, it's explicit in the fruity, fleshy fungi that's coming through, but there's lots of other organisms that are also dwelling there together. And we can see some of the bryophytes, maybe some arthropods and such, but there's also many different types of organisms that are living below ground. And in the montane meadow, similarly, there's likely lots of mycorrhizal fungi that are supporting these plants. And the community feeds back to really be able to support each other. And those factors like environmental filtering or host filtering in the case with symbiosis, where you have a host that has a number of different microbes living at their roots, mycorrhizal fungi and other bacteria and such, the host may have certain characteristics that then filters the whole community that they dwell in symbiosis with. And there's all of these different characteristics of the community. We're not going to go really into this in too much detail, but as we wrap up the section of molecular microbial ecology, I want you to consider some of these things: diversity, the relative abundance of different species that may be in the community, and the structure of the community. These are some things that we're thinking about when we apply these techniques that we'll cover here in today's talk. So one thing I wanted to mention within the context of biotic filtering is this concept of niche overlap. So a niche, or the range of resources that are used, or how an organism meets its needs and makes a living, sometimes there's many organisms that want to colonize a hospitable niche. And when there's overlap there, one species may be outcompeted by another, or driven to extinction. And we can see this here on this plate, where we may have staph microbe growing and penicillium mold growing, and there's a zone of inhibition around the penicillium. So there's been a lot of really fun studies on fungal combat with wood chips or in petri dishes, where you can actually see which is the most competitive fungus of the two or the three, and create these little mock communities and monitor them over time and see who adapts and who perishes, and who coexists. Competitive exclusion, which was proposed by Gauss, that if two species with the same niche coexist, one will be excluded from the community due to this competition. So this led to niche partitioning. And we can see that nicely with these roots of these prairie plants, where some roots are really some plants have really deeply rooted systems, and others are more shallow. So they can share the niche, and some may even acquiesce to the other more competitive species, and figure out ways that it can live and dwell in harmony. Grimes' pyramid is another kind of ecological framework that uses the concept of competition. And I'm going to talk about it just briefly within the context of trade-offs. So if you're really competitive, your growth rate may be really vigorous, and you may grow very fast in favorable environmental conditions. But when you're highly stressed, or there's a lot of disturbance that happened, you may not have the skills or the mechanisms to be able to propel and fuel your growth. So thinking about this in the context of mycorrhizal fungi, there's the fast, more competitive colonizers that are like your weedy species that grow very fast, and some are more competitive than others. And there's a trade-off between this ability to grow really fast in maybe a more stressful or disturbed environment, and you can be the first one there, but then maybe you're less competitive. And similarly with a competition stress trade-off. And how does that translate to fungal decomposition or use of resources? Well, here's one example where we have two food sources. The graph on the left has glucose. It's measuring a really easy to digest, labile, which is easy to break down, carbon compound, glucose. And the ability to degrade glucose goes up with your ability to tolerate drought. But then if you want to look at a very difficult to degrade compound like cellulose or lignin or chitin, as you degrade more of this, it just requires so much energy putting into the degradation, to synthesizing those enzymes, to biodegrade these complex, difficult to degrade recalcitrant compounds. And then you may be more susceptible to a stress like a drying environment or drought. So there's different trade-offs that these organisms make in order to fulfill a particular niche in the community. And these dynamics are really, they're considered intransitive, where it's really a rock, paper, scissor kind of system where you don't know, you can't predict what's going to happen just by knowing what you're going to put out. If you put out rock, you might either get covered by paper or you might bang the scissors and break them. So these dynamics differ depending on who is in the community, how they grow, and what their interactions are. So I have this little flow chart here where some species are very sensitive to disturbance, or they're very susceptible, and they will perish in very severely polluted environments or highly degraded environments. They're super sensitive, and they're the ones that will shrink back and leave a zone of inhibition in their path, in their wake. And then it takes a little bit more energy for this other group, the resistant species. These fungi are the ones that we would consider tolerant. So they have the tools to be able to tolerate the stress. They're not sensitive to it. They're not going to transform it actively, but they can tolerate it. They can sort of hold their ground. But then I'm using this term producer or toxophilic, where these producers are able to generate something like extracellular enzymes or something that is going to outcompete the other strains that are competitive. It's going to produce something, because not only does it have the ability to tolerate the stress, the pollutant, but it also has the metabolic machinery, this enzymatic or metabolic genomic architecture to be able to produce something, synthesize something that's actually going to denature that toxin. So that's when we start thinking about metabolizing something. And then when we put that back into the context I was just talking about with trade-offs, these evolutionary trade-offs, if you're going to produce something and synthesize something that's very expensive, that's going to dismantle a toxin or reduce the threat, that may be so energy intensive that you have a very slow growth rate. So these are just some concepts to think about within the traits of some of the microbes in the community. Oops. So now let's think a little bit more about how we may characterize how sensitive fungi or species may be to environmental factors, because there are these feedbacks between global change and the structure of the fungal community. So this paper came out in 2008 by Steve Allison and Jennifer Martini and a few other folks at the university where I did my PhD, UC Irvine. And it's all about microbial resistance, resilience, and redundancy. And if it's not in the folders for this week, I'm happy to put that paper in. So this lightning bolt stands for some sort of disturbance. So you have a disturbance, and it either has no effect on the microbial community, they're resistant, and their composition stays the same. So how abundant all the species are in the community, that's the composition, or the structure of the community, that stays the same. Or, you know, we think a lot about resilience. it could be that the microbial community composition is altered. It does change, but then it goes back to the original composition. So that would be a resilient community. It's resilient to change. It is affected, but it's a little bit more of a blip. It affects it, but then it goes back to the original composition. So another thing that could happen is that the microbial composition, you know, post-fire or after a severe chemical spill, the composition could be altered and it could remain altered. And in that case, it may require direct inoculation or direct mycorrhizal or microbial inoculation in order to restore the structure of the community to the pre-disturbance state. But I do want to mention that even if the composition remains altered, in certain cases, the functions that are performed by that community may still be there. So there would be different species in the community, but it'll perform the same as the original community. There'll still be nitrogen fixation, there'll still be decomposition. Those rates of process won't change. And that's because similarly in permaculture, when we talk about many elements can be supported by a single many elements support a single function, and many functions can be performed by a single element. It's similar in that case where the functions can be performed by a number of different species that are all nitrogen fixers. Maybe it's just different rhizobia, not the same ones that were there before the disturbance came in. Now it's different microbes, but they're performing the same function. And then the last kind of stable state that could happen after the disturbance is that the composition is different and the processes, the functions are also changed. So this is the different endpoints from a disturbance: resistance, resilience, or redundance, or completely changed. Just today, I submitted an abstract to present at the Ecological Society of America meeting in Salt Lake City in August on my work looking at mycorrhizal fungi and bacteria and fungi after the eruption of Mount St. Helens. So it's interesting looking at that data and thinking about what sorts of land use practices and different experimental manipulations have led to very different microbial and fungal and mycorrhizal communities after this devastating volcanic eruption. So, you know, just thinking about these massive disturbances and what's the successional processes that allows the community to assemble afterwards, and what small changes, like the butterfly effect, what small changes can we make that actually can promote the recovery of entire ecosystems. Because ecosystem functioning is really, it's something that can be characterized in so many different ways, but diversity, the number of species that are there, really plays an important role in the functioning of the ecosystem. Here on the left, I have a high diversity system where all of these circles represent different species. And on the right, there's a fewer number of species. So a high diversity versus a low diversity. And there's been countless papers, or maybe not countless at this point, but many papers that have looked at the links between biodiversity and ecosystem functioning. And we see that with the loss of diversity, we lose a lot of important functions because low diversity systems are more vulnerable to threats and they're less complex in many ways. And there's been a number of studies that have looked at plants, how plants and plant diversity is linked to the functions that the plants perform in the ecosystem. But there's been fewer that have looked at fungi. So that was something that I wanted to look at a little bit, and I'll use some of my research and some of my colleagues' research in order to showcase a few experimental designs and think about how we may use that for formulating testable hypotheses. And in terms of experiments, there's a ton of observational experiments we can do, and also manipulative experiments in order to try to get at mechanisms and executing these experiments and then analyzing the results. And we're not going to really get too much into analysis today. So I wanted to just mention, I'm not able to see the chat. So if anybody has a really pressing question and such, I hope that we can cover it either at the end or someone can give me a little signal so I can address it.
Mia,
Sure.
it's Rebecca just checking in.
Uh-huh.
There has been it's a text box that is on the right side of your
Thank
slides.
You.
And
a text box?
Yeah, it's the color options.
Oh, this thing. Oh, thank you. Okay,
there you go.
Perfect.
All right, great.
Is that better?
That's better.
Oh, good. Thank you for mentioning that. I really appreciate it.
Yeah, thank you.
Okay. All right, so we're moving on in the presentation. We've already knocked out the first thing, and we'll move sort of a little bit more quickly than I like to through the next parts here. But in general, you want to have randomness and replication in your experiments. You really want to make sure that you're randomly selecting things, and sometimes I just throw a stick over my shoulder. Where it lands, that's where I collect a sample. And I'll write this in the methods section of my paper. That was how I randomly collected the place to sample. But it gets more and more complicated the larger your sample size in your experiment is. I like to use the random number generator as well. I can use that in Excel or just kind of Googling that. If I need to try to create some random numbers so that I can really get a sense that my observations are not inherently linked to another. Like for instance, a study that I conducted in China looking at the effect of acid rain and mycorrhizal fungal inoculation and revegetation strategies on soil biogeochemistry and bacterial and fungal communities. The study had some flaws in that the distance from the river and where the plots were laid out were not randomized, that there was a whole treatment group that were just closer to the river than the other treatment group. So I had to really restrict my analysis to account for that. So there's a lot of different ways that you randomize it, and I'll show you a few different techniques like the randomized block design. But you want to have some randomization, some replication, and independence within your experimental design. So one innovation with trying to link plant community diversity with fungal community diversity and ecosystem functioning was looking at leaf litter fungi and the number of plant species in litter. And I use this innovation called the fungal cages. And either I'll do it with window screen and I'll kind of close off, make these little cages and put litter inside. Or I'll use very fine mesh that's 0.22 microns, which allows for the movement of water and nutrients to pass through, but it'll keep the microbes, bacteria, and fungi within. So it'll control the composition in that case. And I can make these little packets, these little fungal cages in a number of different ways. I usually use like a hair iron to seal them along the edge with the nylon mesh. And then I can replicate, have a lot of these little cages, and each one is a sampling unit. So I manipulated the number of plant species in litter in these little cages. Those are ones with window screen. And then I deployed those in these different fragments of coastal sage scrub, which is an ecosystem nearby where I live, to evaluate the effect of habitat fragmentation, like the size of a fragment and the number of plant species in litter and how that would affect decomposition or fungal diversity. And because I had enough replicates, not a huge amount, but I had enough replicates per plant species, I was able to show these patterns where decomposition was linked to greater number of fungal species, and the greater number of plant species harbored greater numbers of fungi. So these links between diversity and ecosystem fungi were evident from this experiment. Some other experimental designs, I do a number of studies that have large geographic scales, like all across the whole Western Hemisphere or globally or continental United States plus Puerto Rico. And each of these sampling plots, the samples are collected the exact same way. So this is just an example of 13 samples that are collected in the same spatial orientation across all of these plots in North America. And one of the things I think is exciting about having a group like this, where there's this motivation and this shared knowledge, is that we can do these large scale experiments and collect samples in the same way. And then we can compare across biomes, across ecosystems, across disturbance types, and see how some of those factors affect fungi and fungal community and function. Another thing that I wanted to mention in considering experimental design is gradients. Gradients are incredibly useful, like for instance, elevation gradients or mountains. Mountains are excellent laboratories for climate change because they're often colder and wetter at the top of the mountain and warmer and drier at the bottom. And along with those gradients in temperature and moisture, there's also gradients in soil organic matter, carbon to nitrogen ratios, pH, as you move uphill. And these are just some results from a study we published in 2018. I thought it was maybe 2016. Yeah, something like that. That date might be wrong, but the paper's in your folder, so you can check that out. That as you move uphill, there's more fungal hyphae at the top of the hill. And this is an elevation gradient in Costa Rica. There's more fungi, like greater biomass, more hyphae at the top of the mountain than at the bottom of the mountain. Fungal diversity changes as well as you move uphill. Higher fungal diversity at the base of the mountain, less diversity at the top of the mountain. So using mountains and using elevation or other gradients is very useful. I have some elevation gradients that I'm working on in California's Sierra Nevada mountain, as well as I do a lot of work in the soil profile. So looking at the depth of soil as another gradient for structuring the fungal community at depth or at the surface. A book chapter that I put out recently looked at restoration, different techniques for indirect, intermediate, and direct restoration, including adding microbes, so microbial inoculation, and specifically mycorrhizal fungi, AMF. And as many of us know, these arbuscular mycorrhizal fungi form symbioses with plant roots. And as they penetrate the plant roots and invaginate the plant root cell membrane, they exchange nutrients and resources with the plants. And they often form these little structures known as arbuscules, which is where we get the term arbuscular mycorrhizal fungi. And I know Taylor's going to cover that a little bit, and we'll cover it more in the course. But I'm just mentioning this as an example of a group that we can target in some of our experiments. So I looked at moving topsoil from place to place. And if I added more topsoil from an ecosystem that was very diverse, would it affect mycorrhizal fungal diversity and plant diversity in these different spots? So here I have these treatments where I have it shown in color, the control, the dusted, just a little sprinkling of soil, or 3 different thicknesses. And I put these treatments together in a randomized block. And this is my experimental design where I had the treatments, they were put together in a block. So they were all represented within one block. And I could have had multiple blocks at each site, but instead I just had one block, I had 3 blocks per site, and then 3 sites throughout the landscape. So here you can see the blocks laid out in the landscape, and I just kind of flipped one around, but typically the treatments would be randomly distributed within the block. So you're incorporating randomness and then also incorporating replication. So at every one site, it's replicated 3 times, and then there's 3 sites. And usually we like to do at least 3. I'd say 4 or 5 replicates is better, but you really don't need that much more than 5 in order to have a robust, statistically sound experimental design. So just like a little bit of results on that. When we added thick topsoil onto these locations, we had a greater diversity of AMF, and specifically the rhizophilic AMF that help protect plants from pathogens. So I just submitted this study to the journal Diversity on Saturday, so hopefully it'll get published and you guys will read it. But yeah, just thinking about how we can set up our experiments in a way that we can then extract that information and try to derive biological meaning. So I'm going to present a little bit of work, a little bit of research in collaboration with Dr. Sydney Glassman, who is at UCR. MycoRenewal is collaborating with some land managers from the Bureau of Land Management, and we've gotten some really generous support from Metabolic Studio, which is an environmental art studio out in Los Angeles. And some of the work we're doing is in collaboration with Sydney Glassman. She is so excited about the work that MycoRenewal is doing, and she really wants to help support this type of research. If you're interested in her on Twitter, she has Fungi Foodie, and also her lab is the Glassman Labs at UCR. We're now moving into our fungi and fire component of the talk. I just want to check the time. Kind of need to wrap it up here pretty soon. So I will just say that it's very difficult to study fire. It's very difficult to study mega fires because even though we may predict which locations are vulnerable to fire, in general, it's hard to predict when they're going to go into flames. So going and collecting samples right before the fire and right after in order to really get a snapshot of how fire affects the community right away is challenging. It's really difficult. So there's a few ways that we're approaching not only going in after the fire and evaluating how the microbes are doing, but also allying with some collaborators in order to examine prescribed fires. One exciting innovation is the pyrocosm. So we can make these small simulated fires that allow us, the amount of fuel that we add is directly correlated to the heat. So we can look at fire severity and examine how fire severity may affect fungi in soil or in litter, and create these manipulative experiments. And we're setting some up at UC Riverside, and also I encourage you to read the bioRxiv paper by Tom Bruns's group in development of this pyrocosm. And if it's not in your folder, I'll put this paper in there as well. So many animals and plants have developed numerous adaptations to fire. The most famous is probably serotiny, that serotinous cones need fire to germinate. We know morels have sclerotia that enables them to persist past fire. The echidna in Australia has an interesting mechanism where he's got long spines, like porcupine spines. They'll just burn down and it protects the actual animal, but the spines will get a lot shorter. So if there's these traits that we see in living organisms, what are the fungal traits or the bacterial adaptations to wildfires? So in our work, we're looking at how the composition of the fungal community feeds back, how their traits, the fungal traits, then influence ecosystem functions like biogeochemical cycling. And one thing that's been kind of fun to do is go out when there is a fire and collect microbial or fungal propagules from the wildfire smoke. So we've gotten a bunch of cultures going from soil and from smoke, and seeing if any, these are fungi and bacteria that are moving through smoke that still are viable. They're still able to grow on a petri dish. So taking a look at those, and then that gives us a culture library for doing some more experimentation and some more interrogating their genomes and see what is it about their genomic architecture that makes them capable of persisting and maintaining their viability in fires. We often see these orange peel fungi, which it's kind of exciting to see these open cup apothecial ascomycete fungi, that there's several different species of orange cup fungi that come up in fires out in the Great Smoky Mountains, here in California and Santa Rosa, different places. So it's interesting. What is it about this feature that allows them to grow? A closely related fungus, Neurospora, is a fire-following fungus, and it's one of the first most weedy species that comes out after a fire. So I'm kind of interested in that in terms of the thick-walled ascospores that all of these species share, but also a lot of the truffles too seem to be very resilient or even seem to love fire. And these fungi may be key to mediating regeneration of ecosystems after these disturbances. So I'm really interested in regeneration phase fungal inoculation post-fire. So this is a picture of the Rim of the World Vista fire that was out in Yosemite. And you can look before the fire and after the fire and see differences just with the naked eye. In many cases, the trees are dead and the giant stumps have burned out in the very severe fires. And also the seedlings that are there, or the spores that are resident in the soil that survived, we can do bioassays with plants to see if you can bait those spores from the soil. And you can see these little ectomycorrhizal root tips from these seedlings that were previously sterile, surface sterilized seeds from the spore bank, from this bioassay. Here's a burnt chaparral ecosystem near where I live in Southern California. So we get inspired as what we can do to try to help restore these ecosystems. So what happens when you add fungi to degraded systems? I'm going to scale back just a little bit near this end of the talk here. You know, typically with mycorrhizal fungi, we see a big increase in plant biomass, like a doubling of biomass when you inoculate with mycorrhizal fungi. And my 2015 meta-analysis showed that inoculation has a positive effect on mycorrhizal colonization, but definitely the source of inoculum matters. So soil from a reference ecosystem, as we see here with the A bar, is more effective than commercial inoculum, at least in my meta-analysis. And that paper from Restoration Ecology is in the drive. And also inoculation has this big effect on plant performance. And it doesn't seem to matter whether it's arbuscular or ectomycorrhizal, where inoculations were done, if it was in the nursery or in the field, or what the inoculum source was. So we have this sense that when we have a landscape that's been affected, maybe especially if the soil is depauperate or has very little remnant mycorrhizal spores that are there, adding those spores could have a real positive effect on promoting revegetation. So here's one of our field sites, this map by Lake Matthews. And I'm just going to show you the use of quadrats and transects. Many of you that have done ecological research have used these techniques. They're just lines where we have experimental plots along the lines. Each one of those window panes are different treatments. And I have plots that are unburned, plots that are burned, and treatments along these transects, these lines. And our experimental design for this project is also in the drive. So each one of these colored boxes represents a different treatment, whether we added mycorrhizal spores or just soil or native seeds or some sort of combination. And I'm just going to flip through these. I'll put all these slides in the drive, but basically setting up your experimental design helps you come to these inferences that ash depth does correlate with burn severity. And we do see that in our data. It also, the severity of the fire affects soil pH. So that's another thing that we can say with certainty based on our experimental design. And fire severity also has an effect on the respiration of microbial communities. So we can use these quadrats, which are boxes, 1 meter square quadrat that I just made from PVC, and use it to measure ash depth or ash cover. And I created a little the amount of bare ground, ash, vegetation and such, and I just record all that data so I can look at it. And oftentimes with my quadrats, I'll stretch a netting over it, so then I can look at plant community composition at each one of these point intercepts within the quadrat, when any of these intersects of the netting. So just kind of moving through a little quickly here. We have soil inoculum and spore inoculum, and I extracted spores from the soil using a sucrose density centrifugation. And I'm also working on different lower tech techniques for extracting mycorrhizal spores from soil, like with wet sieving. So as I develop and document more of these protocols, I'm really excited about making them available to folks. Some of our experimental plots, and just linking how disturbance like fire, the frequency and the severity of the fire may affect fungi and plants, but inoculation could also help restore the vegetation community and the biogeochemical cycling. So we're just here, just about out of time. I'll just move through this microbial ecology piece pretty quickly. Let me see. I'm at 50 minutes now. I'm wondering if maybe I should wrap up and not cover the microbial ecology if we want to take some time for questions, or I could take a couple of minutes just to cover microbial ecology. What do you guys think?
We could always save kind of like group questions for the very end, both after your and my presentation.
Okay.
I feel good with that. If people want to stay on for a little bit longer, we can have open space for questioning if you want to go over this portion, Mia.
Okay, I'll try to do it in just a couple of minutes then. So as I mentioned at the beginning, microbes are found in every environment, and this concept of microbial infallibility, that every molecule can be used by a microbe. So we could sample the organism, and here I show Rhizopogon olivatio tinctus, which is a fire-following fungus. We find it, this truffle, it's from the Boletales, so it's a bolete truffle. We find it, or a hypogeous fungus. It grows below the surface, doesn't have a fruit body that comes above ground, so it's not epigeous. So we could find these fungi fruiting post-fire, but when we use molecular techniques to characterize it, we find that it is highly dominant after the fires. Not only do we just find a couple of fruits, but we use molecular techniques and we show that it is a key player at post-fire. Not only in pine forests, but also down in these chaparral sage scrub ecosystems as well. So not just sampling the species or sampling the organism, this microbial ecology approach is really focused on sampling the environment. And just in the next couple of slides, I'll mention that typically I would take my sample and extract DNA from it by breaking open the cells and isolating the DNA. And then I conduct this polymerase chain reaction in order to make many copies of the DNA for the region that I'm interested in. So I'll go over that again here. So let's say I'm extracting DNA from soil. I will then use different primer sets like the 16S set for bacteria, the ITS for fungi, or Wanda AML2 for AMF, and I'll target a particular part of the DNA that I'm finding in the soil. And the soil represents my whole community. So I'm not just getting the DNA from one organism from a single soil, I'm getting all of the, let's say I'm using ITS for fungi, I'm getting sequences for all of the different fungi that might be in a 1/4 gram of soil. So that's what I use to characterize the community. And I sequence it on the Illumina MySeq, which is an instrument that we have at our genomics core at UC Riverside where I work. And then I can use it to characterize the soil metagenome, the fungal community, or the AMF community. And I'll just go through that again because I want to just mention that I can extract DNA from root tips, like arbuscular mycorrhizal fungi or ectomycorrhizal fungi, fungal root tips. I can extract it from a single mushroom or a whole soil, or what I've been doing is looking at the microbiomes of many different things. And I'm happy to talk with folks more about that if you're interested. But I target the region of interest, so a gene or a region that is found within that particular target group. And then I use gel electrophoresis to visualize that I actually got those fragments to amplify with my PCR reaction. And then I do a magnetic bead purification with these weird little magnetic beads to purify my sample. And then I do it again, because what I want to do is add a little molecular barcode onto all the fragments from one sample to be able to then show that all of those hundreds of thousands of fungi that I found from that 1/4 gram of soil, that all of those now are tagged with a little barcode. So then I can look at many different samples. Sometimes I'll put 400 samples in one run. I'll get 25 million reads. And once they have these molecular barcode identifiers, then I'm able to say, oh, these ones came from Hawaii and these were from Alaska. And I can compare the fungal community in those different samples. And I do the sequencing and the bioinformatics. And then I can visualize the statistic or visualize the community with some sort of, this is called an ordination. So some sort of space where, if you can see the continent in the bottom right inset map, the light green samples are from the East Coast and the reds are from sort of the Pacific Coast. And you can see, oh wow, geography really structures fungal communities. And then just to end, I'll say there's a paper in the drive about Fungild. It's a database that takes the sequences and matches them to different functional groups. So if they're pathogens or symbionts or decomposers, and it goes to different guilds and trophic modes. So I'm finding it really exciting to be able to look at a landscape like this that is very affected by fire and be able to evaluate over time using these molecular techniques what's happening to the communities that are assembling after that disturbance, and try to see, are they resistant to disturbance like a lot of bacteria are? Are they resilient, or do we have functionally redundant communities, or actually are they so sensitive that the functions aren't able to be restored without microbial inoculation? And with that, I'm happy to wrap up. Thanks, everyone.
Yay, thank you so much, Mia.
Of course, thank you. See if I can get out of this. Great. Check out the chat.
Yeah, there were some questions for you in the chat box.
Okay.
I'm gonna go ahead and segue into my presentation, and then maybe afterwards we can hold space for questions for both of us. Does that sound good
great.
for you?
Yeah, that's great. Thank you so much.
Yeah, thank you. And if you don't mind, hopping off the screen share.
Oh, did I not do that yet? Okay, great. Stop share. All right. Thank you.
Yay. Thank you, Mia. All right. And then we'll just move into mycofiltration and fungi in wet environments. We'll take just like a little bit of a breather to kind of transition the energy into water. Yeah, once again, I just want to say thank you, Mia, for all the work that you do and all the research that you do. It's so inspiring and incredible. And let me get up my screen share, and then I'll introduce a little bit about myself. Then we'll move into water. Okay, screen share. How's everyone doing? Good? Wonderful. You guys ready to learn about some mycofiltration? Okay, let's see. Can everybody see this presentation as well? Is it up? Okay, wonderful. Trying to figure this out. Cool. Well, so hi, I'm Taylor, and I am a citizen science mycologist based in Northern California. And my focus of work right now is learning as much as I can about mycoremediation and applying mycoremediation techniques. And I'm definitely coming from more of a grassroots perspective, which is cool because Mia laid down a lot of beautiful technical like laboratory approaches, and I'm going to be talking a lot about some more grassroots, low tech, low cost, DIY ways to approach mycoremediation. In specific, we're talking about mycofiltration here. And then I'm going to move into fungi's role in preventing soil erosion as well. So jump right into it, just to graze over about water. And I do want to say my PowerPoint presentation is a little bit wordy. That's mostly just to provide you guys with all the information. I will be posting my PowerPoint to the Google Docs so that you can access it, but I'll just kind of flow through it. Don't feel pressured to read all the words because it's a little bit wordy. So yes, water. Water is the biological basis of life. Without water, we wouldn't be here. We are, you know, what I like to say, bipedal sacks of saline solution ourselves as humans. And as we all probably know, fungi are inextricably reliant on water. The presence of water allows for the fruiting of mushrooms and the growth of mushrooms and all life. And water is just a very interesting molecule within itself. It's a polar molecule and it has a really interesting bend in it. And since it's the biological basis of life, Earth's hydrology, the hydrological system, is one of the largest governing forces on our planet. And some fundamentals of mycofiltration. Mycofiltration is a subsect of mycoremediation, and the term mycofiltration was actually coined by Dr. Paul Stamets. I'm sure you all probably have heard his name before. And he came up with this, what he calls a biotechnology, and it's been utilized in many, many different ways since then. He has a lot of really amazing research on mycofiltration. So pretty much within mycofiltration, we are focusing on the mycelium. We're focusing on the mycelial web, the mycelial mat, as a living biological membrane that the water is going to be moving through. And this is going to sequester chemicals and heavy metals, remediate chemicals, and degrade biological pathogens through the extracellular enzymes that the mycelium is producing. And this tackles all forms of polluted water, from stormwater, gray water, eutrophied systems, polluted water bodies, anything you can think of. And so what we're focusing on here, yes, is these very dense, interconnected webs of mycelium. And really, when you look at the different levels of mycelium in these pictures, you can see how they form these mats, these dense biological membranes that water can move through. And you know, why fungi? It's an earth-based approach, using things that already exist in nature. We know of fungi's incredible ability to remediate, and we've discovered these possibilities. So why not take fungi and form homes for them to be able to filter water through? As well as our knowledge and ease of cultivation. This makes our knowledge of cultivation makes mycofiltration possible to be used in studies. And many of our pollutant sources of water pollution are called non-point, which means it's very hard to track them. They come from a lot of different areas, and based on water's mass distribution, it's hard to track exactly where these pollutants are coming from. And like I mentioned, fungi, the mycelium, are producing these extracellular enzymes that can break down these chemical and biological pollutants that are found in the water through some mechanisms that I'll speak on later. As well as fungi work in synergy with many forms of biological life, as well as the bacteria and microbial communities that Mia were talking about, to enhance the activity of degrading these pollutants and these compounds. So this is a little bit of the going in depth about how these mechanisms are working for chemical pollutant degradation. And saprotrophic fungi are used in mycofiltration because of their ease of cultivation. I think Willoughby talked a bit about that, that it's been really difficult for us to cultivate mycorrhizal fungi. So we're using saprotrophic fungi in here in these mycofilters that I'll talk a lot about how to make them and what they look like later. So saprotrophic fungi have these non-specific lignin degrading enzymes. And what's fascinating about all of this is because of fungi's innate ability to break down lignin, these woody compounds, they also, these enzymes have the ability to break down what we call polycyclic aromatic hydrocarbons, PAHs. And as you can see here, they have a lot of these the presence of aromatic rings and hydrocarbon bonds. And so fungi breaking down lignin, they can also tackle these PAHs, these complex molecules. And that's pretty much the basis behind what's happening molecularly in fungi's ability to break down these hydrocarbon pollutants. Danielle, next session, will be going much more in depth about mycoremediation and toxicology. And so I'm really just giving like an overview about what's happening kind of behind the scenes in these processes. And then there's two different, we've kind of separated the saprotrophic fungi into two different types of rotters. There's the brown rot fungi and the white rot fungi. In mycofiltration, we've been focusing much more on the white rot fungi for their ability to produce specific lignin degrading compounds. And you can see the remnants of the white rot fungi are B right here. They leave behind these white fibrous materials. And these fungi have been found to, that they're extremely efficient in breaking down these polycyclic aromatic hydrocarbons. And something I've been thinking about throughout my work and research through this is if we were able to utilize both brown rot and white rot fungi, they produce different suites of digestive enzymes. So the more diversity in the enzymes, in the enzymatic complexes, maybe the more efficacy we could have in breaking down more complex pollutants. And heavy metals as well. These fungi have the ability to sequester heavy metals, as many of you have heard before, through an ion exchange mechanism on the mycelial surface. So the mycelium can both bioabsorb the heavy metals through their permeable mycelial surface, or the metals themselves can just bond and bind to the mycelium without being absorbed. And a process called chelation happens where the heavy metal becomes stable or inert, or it becomes in a non-toxic form in the environment. And then I'll speak a little bit more on that later as well. Yes, and when these sequestering properties happen, when the mycelium are sequestering the heavy metals, they concentrate them in their fruit bodies. And so this is a way to take heavy metals out of the ecosystem, out of the water, out of the soil, and concentrate them in their fruit bodies, which will then have to be removed and treated off-site or ex situ. And there's a lot of different conversations going on around how we can actually do that efficiently, and what it means to really treat these heavy metal laden mushrooms. Different species of fungi target different pollutants. And so here is a list that I borrowed off of Leela Darwish's Earth Repair page, just to show that based on what pollutant you are targeting, it's really going to depend on what fungal species that you choose. So a site analysis is very important to understand what constituent or what compound you are targeting. And based off of that, there's been a lot of research done to show that these different species of fungi can actually target different pollutants and have better sequestration abilities of different heavy metals, just based on their the fungi's own personal constitution and what enzymes they're creating and their capabilities. As I've been reading through this, there's also been a lot of talk on our ability to train these fungal species. So when they are contained in a petri dish, to expose the mycelium, the growing fungi in this controlled container, to the specific pollutant that you're targeting, so that the fungi can adapt to that specific pollutant and actually create the enzymes that it needs to break down that pollutant and use it for nutrition. So that's something that's called like training the fungal species to be able to target that specific pollutant. And then once that fungal species is trained to be able to digest that pollutant, you can then take that and proliferate that species of fungi and then apply them in the environment, and they'll already have the specific enzyme cocktail to be able to target that pollutant. Really speaks to their adaptability and efficacy. And this is a little bit mycorrhizal fungi are filters too. In these filter biotechnologies that I'll soon show you guys, we use saprotrophic fungi for their ease of cultivation, but we can't forget the powerful abilities of the mycorrhizal fungi and the filtration roles that they're playing in their environment. They create deep networks with the roots of plants, and as the water percolates through the soil, they're acting as a natural ecological filtration system, as well as creating healthier plants, healthier and more resilient overall ecosystems that are going to as well help in the natural ecological filtration of an environment. And ectomycorrhizal fungi can also have been shown to also sequester heavy metals and pollutants, and so the mycorrhizal fungi are also doing this role in the ecosystem where they live. And so now I'm going to go a little bit into making mycofilters. Like I said, I come from a grassroots approach of making these biotechnologies, super DIY, low tech, low scale. That's what really I'm all about, is making these approaches approachable and adaptable and really available for anyone to use and understand. So a couple approaches that have been practiced in different studies, both in both case studies and just grassroots approaches, are myco bales, when you just take an entire straw bale and you inoculate that straw bale and then place it in a creek or stream or river. Mycofilters, which are inoculated burlap sacks, which I'll show you guys some of my own. Mycofiltration beds, which are kind of like biological buffers. You just lay down a lot of organic material and inoculate that organic material and just allow the natural current of water to flow through those. As well as drip systems, which are used in a lot of eco-technologies where the water is gravitationally put through an inoculated substrate of some sort, whether it's contained or it's a bale or a sack, whatever it might be, using gravity. And then it percolates from there. I have a lot of thoughts about these types of drip systems being used in home scale gray water systems. And how that might work. So here's a couple examples. In the top left, we can see a lot of the burlap sack mycofilters used. They're being staked down into the ground, and then the water flow is just passing through them. You can also see here that it's really important for the water to be fully flowing through the sacks, but as well as for them to have air. Fungi are aerobic organisms just like us. They require oxygen to breathe and grow. There's a couple other like burlap sack examples, as well as a little bit more complex systems that are using a lot of different kingdoms that are not just fungi, but using a lot of different layers and a lot of different kingdoms in their filtration abilities. So like I said, I'm all about low-tech, low-cost DIY methods, making it affordable, accessible, and approachable. This is also a relatively new and emerging biotechnology. We're still in a huge experimentation phase when it comes to figuring out what works and really how to do it the best. There's a lot of studies being done, more like laboratory-based studies, but there's also a lot of citizen science approaches being done, and just really observing how these are working in our environment. Okay, and all of that being said, there are things to think about when approaching any of these mycoremediation strategies. You don't just want to go willy-nilly into it because you're dealing with a living organism that can, you can go into something with the best of intention, and without knowing what you're doing or having a foundational understanding, you can actually do more harm than you can good. So that being said, mycofiltration needs to be site specific. So listen to the lands. Like, what is the land saying? How are you approaching this project? How does the water move through the land? What environmental conditions are present? Like I said, how does the water move from the site? Where does it come from? Where does it go? What source is it coming from? What contaminants are you targeting? So based on where your land is, are you trying to remediate pesticides? Are you living downstream of a cow pasture and are afraid that there's E. coli present in the water? So you're trying to approach a more bacterial pathogen type of remediation. Or are you living downstream from a mine that has heavy metals present? Just really paying attention to the and what's present on your site. Based on that, does the burlap sacks or does something more detailed make more sense? And how am I going to maintenance my filters? Since we are using living biological organisms like fungi, oops, I didn't want to do that. Since we're using biological organisms like fungi that are meant to break down and degrade organic matter, these filters don't last forever. So making sure that you're present and you're able to maintenance these filters once the mycelium start eating through them. And then soil water tests. This is sometimes the most limiting variable when it comes to these like DIY mycofiltration experiments, but it's really, really important if you have the capability to get soil water tests, both pre-experiment or pre-filtration and post-filtration, and at many points throughout the process, because this is really where the empirical evidence is going to come from to show, hey, yes, this stuff is actually working. And you can be a huge contributor to the scientific basis of knowledge that we have around such a new and emerging application of mycoremediation, as well as if it's not on your land, how are you like engaging the community around you? Are you telling people about this, or are you telling local municipalities? Are you honoring the land that you're implementing these types of approaches to? I get consent and permission from the land you're working on, just like with all bioremediation, but mycofiltration as well. And then you get to show people how incredible this technology is and how it's working. And when I say technology, I really mean, you know, the fungi is really doing the work. We're just using them in a way that makes sense. So this is a really important part about the type of sterilization you're going to use. I've done experiments with mycofilters that have used no substrate sterilization, but there are really easy, low-cost, low-tech ways of sterilizing substrate via hydrogen peroxide soak, which is just a 1-to-10 ratio, 1 bottle of hydrogen peroxide to 10 parts water, and then soaking your substrate in that, as well as a cold water fermentation, which is where you submerge your substrate under the water and create an anaerobic environment. So most of the aerobic bacteria die, or the aerobic contaminants die. And then when you pull that substrate out, then it's an aerobic environment and all the anaerobic bacteria die. So it's a really cool way to do a grassroots way of sterilizing. You can also conventionally boil or autoclave your substrate, but when working with these burlap sacks, we're using a lot of substrate. So it's really nice when you can approach sterilization methods with these low tech, low costs. That being said, there has been studies that show that the synergetic activity of fungi and the microbes present actually have a better ability to target some of these pollutants and constituents, which is why I have attempted to do mycofilters without substrate sterilization. However, it's nice to sterilize because it's giving your fungi that you will grow on the substrate a nice even playing field to grow, a nice clean slate to grow. And then when you place your mycofilters in situ in the environment, it's inevitable that microbial communities will begin to come in and synergize with these fungi. And as always, diversity is resilience. So this is a approach of mycofiltration that I have experimented with. And this is the burlap sack mycofilter, something very easy and approachable that you can research further and do at home. But I'm going to go into some studies that I've done with it. This is a personal project that I did at home, and we use no sterilization for this specific project. We use shiitake, oyster, and king stropharia. Those are the species that we use based on where we were going to put the filters. And here I just used old myceliated blocks from Far West Fungi, which is a commercial mushroom farm down in Santa Cruz, as well as I have a king stropharia bed that I just took the mycelium straight out of that bed and used it to grow on the substrate. So here are some of our materials that we use. As you can see, it's pretty simple. We have rice straw and burlap sacks that we got for free from a local chocolate factory. We have some mixed hardwood chips consisting of willow, oak, and fruit trees. The old myceliated blocks, as you can see there, those are the oyster ones, those big white ones. And then a water source. And this is kind of a step-by-step process of how we constructed these mycofilters. We broke up the myceliated blocks in a mixture of 50-50 mixture of the straw and wood chips. And then we mixed that up and soaked it and saturated it nicely with water. And then we filled these burlap sack bags. And we made about 4 of each kind. And this is mostly just to use in the community, to put in stormwater drains and to use on my personal property for different things. And a little bit of our results. So we let them grow and checked regularly. Unfortunately, we didn't make them at the best time because about a week after we started letting them grow, we got a couple frosts, and that definitely stunted the mycelial growth in the bags. There was a little bit of mold contamination from the non-sterile approach of not using any sterilization methods for our substrate. And the mycelium from the grow blocks that we got from Far West Fungi are definitely not as vigorous as fresh, newly growing mycelium. However, despite all of that, we still got growth. And those are some things I would approach differently next time. Definitely using a sterilization approach and more viable mycelium for that, and letting them grow in a more favorable environment so that we could get mycelial growth faster. But it's always an experiment, and we are always learning. And with the ones that grew nicely, these are some of the implementations. I talked to the local municipalities in Sebastopol, which is where I live, and really tried to engage the community members as much as possible. And in the top left, you can see I put an oyster, a pleurotus mycofilter in a storm drain. This is I was just observing how much petroleum runoff was coming from the vehicular traffic around that area during the rainstorms, and oyster mushroom has a very potent ability to break down those PAHs, those polycyclic aromatic hydrocarbons. So that's why I chose oyster for that. The underneath that is as well as an oyster mycofilter that I just put on the storm drain that's in my driveway, so that when oil or runoff from our cars, drips on the ground, that there's a biological buffer so that that runoff isn't going directly into the storm drain. And then on the right is a couple of the Stropharia rugoso-annulata, the king Stropharia mycofilters, that there's a lot of small scale livestock on the street that I live on, and a lot of contaminants, biological contaminants from livestock waste that's often a problem with livestock and cattle and herding animals. So I chose the Stropharia for that because Stropharia is known to target those biologically active pathogens. So yeah, that's a little project that I did about 2 months ago. And so I've been watching the mycofilters grow, and there's some really exciting results. Another mycofiltration study I want to talk about is I work with Bay Area Applied Mycology. We're a nonprofit organization based in the Bay Area that focuses on mycoremediation initiatives and projects around the Bay Area. And this is a more large-scale approach to a mycofiltration implementation, also using the king Stropharia, the Stropharia rugoso-annulata. I was not personally part of this project. This happened back in 2012, but BAM, for short, teamed up with the East Bay Municipality Utility District and implemented a whole approach to cleansing their water downstream of a large cattle holding area. The people in the area, or Edmund, was concerned that due to the presence of the cattle, that there were going to be some biological and coliform bacteria from the cattle waste that were running downstream, which leads into water reservoirs that were supplying drinking water to the greater East Bay. Edmund uses chloramine, the city uses chloramine to eliminate harmful bacteria and protozoa. So we thought, well, if there's a more eco-friendly way of approaching water sterilization, and we can meet Edmund's standards by using these mycofilters, maybe there could be a supplement for using the chloramine and using fungi instead. So I'm just going to run through that. This was in Duffel Meadow in Orinda, California. This is really an example of like a large scale approach to implementing a mycofiltration strategy. So we did a cold water ferment as the sterilization method. As you can see on the right, all of the straw and the substrate being submerged in water, and then mixed Stropharia spawn in the sterilized substrate at a ratio of 1 to 10 to 1 to 20. So a lot of spawn. As you can see, about 3 weeks after, we're getting these amazing presence of what are called rhizomorphs. They're just like long, more root-like conglomerations of hyphae that are specific to the king Stropharia. They have these rhizomorphic structures. And so you can really see that they're beginning to dominate the straw. Okay, we can go. And so we blended that at a ratio of 1 to 1 with slightly aged wood chips. The myceliated straw we put with wood chips and then packed into burlap sacks. And the burlap sacks were sewn shut, and we had a lovely group of volunteers come out to make this happen. I see a couple of our board members in there, Sean and Mino. Shout out to them. I think Sean is actually on tonight, so hi, Sean, if you're there. And this was the implementation that they did. So there were over 72 total bags that were installed in 2 grids. The bags, after they were inoculated, they were pounded in with these wooden stakes. And this is the main stream of water. As you can see in the little gully, the water was coming from the cattle holding area, coming down this gully, and the Stropharia bags were put right in the water flow. About 3 weeks after the installation, as you can see, there's this beautiful mycelium growing on the bags, which is a good sign that the bags are doing their work. And then not too long after, about 2 months later, we were getting fruitings of these king Stropharia. And if you've never seen king Stropharia, this is them. They're so beautiful and amazing and edible and delicious. And these were growing right out of the burlap sacks. And so it was a really, really exciting study. And this is following a lot of Paul Stamets' foundational directions and a lot of his studies for, he's done some studies on king Stropharia and their mycelium's ability to remediate the biological pathogens out of the water, specifically E. coli. So we went with his studies in this implementation. However, we were not, we didn't get a lot of our own studies or our own samples tested. So this is really just kind of like an overview of the implementation. And there wasn't a lot of follow-up on the studies, but it seemed to be really good. And after the implementation of these mycofilters, this is a really exciting part for me, because you're using biological life to make this filtration process happen. And once the mycelium starts growing, it really becomes this self-regulating ecosystem. When you put these filters in the environment, they attract bacteria and other microbial communities, which attracts insects and birds and other forms of life. Eventually, the substrate will decompose, and it will just sink back into the earth. And you are kind of engaging in an active reciprocity here. You're giving back biological life, and you're giving back fungi to the environment. And as that happens, it's going to build the soil and reduce erosion and begin to fix nutrients. It's really just like implementing more biological life back into the system. And that's a great part of these mycofilters, because they just go right back in to the cycle. Okay. And so that's a very foundational overview of mycofiltration with a couple real world examples. This is a very vast and ever increasing in vastness field. And I highly recommend to read some of the papers and the resources that I had posted for a little bit more constructive studies that have a lot of really beautiful information in them. And here's a list, but these are all found on the homework assignment. And if there's any missing, I will be more than happy to post all of them. There's some really great BAM study in full. You can read up on a little bit more of the details. That's on our website, as well as Radical Mycology has some really great. Peter McCoy's book, Radical Mycology, has some amazing references for mycofiltration. Okay, I know that was a lot, so thank you guys for holding in there. And now I'm going to go a little bit into fungi's role in preventing soil erosion, which is, soil erosion is a huge topic right now, a huge topic of discussion, especially in our agricultural systems. We're losing a lot of viable topsoil, and it's just a really big issue for both our agricultural system, for overall ecological health, for climate resilience. As you probably know, it all starts in the soil. Soil is really our holding fabric for all ecological functions. And here's two quotes that I really love. To talk of any ecology or its protection is to speak of soil, precious in its connection to the web of life. And that's from Peter McCoy's book. And then Wendell Berry says, the soil is the great connector of our lives, the source and the destination of all. I'm having a little trouble with these slides. Can everyone see the slides good? Is there any blockages? Everything is good? Okay, awesome. And yeah, just homage to the worlds beneath our feet. It's not often that we sink our consciousness down into the soil and really remember how much is happening down there. All so much of Earth's functions come from the soil. Specifically, we're going to be talking about the rhizosphere. And the rhizosphere is the region of soil that contains the roots and associated soil microorganisms that associate with the roots. And it's also known as the root microbiome, these communities of microorganisms that are interfacing with these roots. And all of this activity is being carried in the darkness. You know, we often think of the sun, that is the facilitator of all of terrestrial life, which it is, of course. But just really thinking about that, so much activity is occurring in the darkness of the soil. It's kind of that bridge between the light and dark worlds. And all of the cryptic organisms that are carrying out the ecological functions in the soil, from nutrient cycling, carbon sequestration, the decomposition of organic matter, the microbiotic community, the checks and balances that are happening there that Mia spoke extensively about, ecological water filtration and remediation that's occurring in the soil continuously from the fungi and the microbes. As well as aiding in plant growth, which makes for overall ecosystem resilience. And really, when we think of soil ecology, we can think of what all ecosystems need, which is shelter, air, food, and water to live. The communities in the soil are no different than the communities on land. They need those basic resources to thrive. Amazing diagram powers culture with complexity and the interconnection of the soil food web and the soil biome. And as you can see, this is a very regenerative system. What enters the system is being exchanged within the system itself. There's internal processes that retain and recycle the resources that are entering the system. So it's the soil food web is a regenerative system within itself. And you can see there's so many characters and so many players taking part in this incredible world right beneath our feet. And here are some staggering statistics that I thought it was just important to share, just showing how much of a crisis, an issue, the loss of topsoil is. I'll just read through a couple. About half of the topsoil on the planet has been lost in the past 150 years. There is this statistic that's floating around now that's saying, it's a bit concerning and really intense, but it's estimated that we have about 60 years of agriculturally viable topsoil left. And that's just like really hits hard. And each year, an estimated 24 billion tons of fertile soil are lost due to erosion. For a little bit of a timeline, it takes approximately 500 years to replace 25 millimeters or 1 inch of biologically active topsoil through natural biological processes. And in the US, soil used in conventional agricultural practices, I just went ahead, is eroding 10 times faster than it can be replenished. So yeah, we're dealing with the soil crisis right now, and how can we be roles in that, be players in that, in the regeneration of our soils. This is I'm gonna just check time. Okay, I'm gonna start rolling through this a little bit. This is just a general progression of soil loss and erosion through conventional clear cutting practices, as well as conventional agricultural practices. Just talking about the loss of vegetation and the disturbance of heavy machinery, how it compacts aerated and aggregated soils, which leads to the loss of organic material that these microbial communities and the fungi need to live. There's a loss of moisture, there's a loss of ecosystem regulated microclimate, which I'll talk a little bit about later. But yeah, just this successional loss of topsoil based on our conventional ways of clear cutting and agriculture. It's a cascading process, and everything is very interconnected. And with the loss of the microbial communities keeping the soil rich and biologically active, that's what really leads to the susceptibility of erosion. Once the soil is no longer aggregated, wind and water runoff can just really take this soil matter, and if there's nothing holding it in place, then it's susceptible to erosion going into our downstream aquatic environments. So let's ally with fungi to create these resilient soil food webs in these systems. Fungal hyphae will physically bind the soil. When you think of the interconnected web of mycelial networks, you can really start to imagine how those are working in the ground, as this web that's literally a kind of like a holding fabric for the soil itself, as well as the fungi are converting hard to digest organic material into more bioavailable forms for the soil microbial communities to use as a source of food. That's mainly the role of the saprotrophic fungi. And then the endo and ecto mycorrhizal fungi are playing a huge role in plant health and root health, and overall ecosystem resilience. As you all know, roots in the soil can literally act as a holding fabric in the soil. So the mycorrhizal fungi play a huge role in making sure those root systems are healthy and alive and vibrant. They also play an essential role in the carbon, nitrogen, and phosphorus cycles, which was talked about last week. Brendan talked about all those cycles and the fungi's role within them. And they're everywhere too. They can be found in every environment, from lichens in Antarctica to these beautiful microbial crusts in the desert. And they're incredibly resilient. They can be found in a wide array. And what fungi are really doing is producing a compound called glomalin, which is such an important part in any conversation about soil. And it's something that is still a little bit under the radar that I really want to illuminate in this talk, as well as phenomena like the soil sponge and how that's contributing to greater water holding capacity and greater water retention in the soil. And like I said, mycorrhizal fungi playing such a huge role in the overall ecosystem health and resilience, and the health of plant communities that are holding the soil together. So glomalin is an incredible carbon-rich glycoprotein that is exuded by the AM fungi, the arbuscular mycorrhizal fungi, and it literally acts as a soil glue. It creates what's called soil aggregates, which allows the soil to maintain structure, as well as it allows it to be aerated, and it stabilizes the soil as well. So when you have really healthy, stabilized soil, it's what a lot of people call tilth. And it's this smooth, nicely aggregated soil. It's not just a bunch of sand and dirt. It's like that thick, brown, yummy, rich soil. And glomalin itself is, the compound itself is 40% carbon. And so a staggering statistic that I learned recently is that it constitutes for about 1/3 of the world's carbon storage. So 1/3 of the world's carbon storage is actually held within glomalin within the soils. And it's really glomalin doing a lot of this work of carbon storage and sequestration, which is incredible. As well as well-aggregated soil has increased aeration and water holding capacity, which creates better habitat for the soil microbes to be able to do their thing. And soil with the presence of glomalin is often referred to as stabilized soil. The saprotrophic fungi's role in this is they are kind of like the frontliners in the soil food web. So they are taking the organic material, the organic matter, and breaking it down into bioavailable compounds that can then be utilized by some of the secondary consumers in the soil food web. So the saprotrophic fungi would be referred to as the primary consumers, taking that organic matter, breaking it down, and then from there it goes into the rest of the soil food web and gets recycled back. So saprotrophic fungi play a huge role in the regulation of that soil food web. And then we get into the mycorrhizosphere, so where all the mycorrhizal fungi are doing their work. And we've spoken a bit on mycorrhizal associations, so I won't go too much into that, but the arbuscular mycorrhizal fungi are associating with about 98% of all plant species. This is really helping for healthy and resilient plant communities and ecosystems. It helps with plant tolerance, expanding the root systems of plants and really creating that web that's acting as a holding fabric for the soil. It's creating underground highways of nutrient and resource transfer. And these networks overall build soil structure. They're helping with plant diversity, and as we know, diversity equals resilience. So without the presence of these mycorrhizal fungi in the rhizosphere, we would not nearly have the diversity and resilience of plant communities that we have. With the presence of healthy plant communities comes microclimate regulation. The presence of trees and plants and healthy trees in a forest actually create their own climates, which can generate moisture and create this feedback loop where the forest itself is calling the rain and holding lots of moisture in that cycle. This is a little chart that I found from one of the papers that I posted in the resources, and this just goes into a lot of the different roles that soil fungi are playing, the different biodiversity of soil fungi. I don't really think I have enough time to go into the details of this chart, but it's pretty straightforward if you look at it. It's the role that fungi are playing, their services, some soil bioindicators, and fungal plant microbe interactions, what's all happening in there. And the paper also talks much more about it, but it just really goes to show all of the ecological roles that fungi are playing within the soil. And they really help with a phenomena called a soil sponge phenomena, which is healthy soils being able to hold and retain water in a much more efficient way. Like if you had a sponge that is already moist, if you add water to that sponge, it's going to be able to soak up that water much more efficiently. But if you have like a dry decant sponge and you put water on that, if you pour water on it, it might just run right off the surface. And so that pretty much goes into the soil sponge phenomena, that healthy microbially rich soils can have a much greater water holding capacity, and which is a huge basis of what glomalin serves. Glomalin building that soil aggregation, that soil aeration, and really doing a lot of that work in the soil. This is a little bit about the microclimate phenomena I was talking about. And there's a Hawaiian proverb that I'm not going to try to direct translate for thousands and thousands of years. And ultimately what happens here is the evapotranspiration that's happening through the stomata of the plant. Plants are always regulating themselves, so there's water vapor actually being released from the trees and the plants themselves in forest ecosystems, and this is rising up to the surface and creating water vapor in the air. And then an amazing role fungi play in this is the spores that macrofungi will release can drift up into the air, as well as other compounds that are released by plants called phytochemicals, and they act as hygroscopic nuclei in the air, or condensation nuclei they're called. And these larger macromolecules are attracting the water vapor that's in the air and condensing them to actually form clouds that will eventually turn into rain and fall as rain. So it's this incredible feedback loop, and these what they call atmospheric pumps that's creating the rain that forests need. So the forests themselves are creating the environments. Fungi play such incredible roles in that. Myco-friendly farming, so regenerative agriculture is a huge variable in a lot of the soil crisis that we're dealing with now, and we really need to start shifting to more regenerative agricultural practices. Soil tillage can destroy these fragile mycorrhizal networks that take many years to form, as well as our overuse of fertilizers. Over-fertilized plants can actually discourage the natural formation of mycorrhizal fungi. A plant that is saturated in nutrients won't necessarily need to create these mycorrhizal relationships, and it can actually lead to a huge loss in soil fungal diversity. This leads to loss of soil glomalin, and it decreases soil permeability and porosity. And the good thing is we have very easy methods of approaching some myco-friendly farming techniques, just not using tilling, adding fungi back into the soil via arbuscular mycorrhizal inoculum, which can be found conventionally now. And this provides the plants with natural immunity boosters, and it provides nutrients, which ultimately leads to less need for added fertilizers and herbicides and pesticides. Mycorrhizal fungi are creating an immune system for the plants, so that we don't need to add so many inputs to keep a natural system regulated. And there's much more to speak on there. I'm really just giving an overview on these things. And the last little bit I'm going to talk about, I know we've just reached past time, but this last little bit I think is really important, is the approach of mycoforestry, adding native saprotrophic and mycorrhizal fungi back into forest ecosystems. This is contributing biomass back into the ecosystem, adding to the soil resilience by adding mycorrhizal fungi back into the ecosystem. It can lead to carbon sequestration and reducing fire fuel loads. So a lot of places are felling trees, and all these trees are laying on the ground. And I think someone mentioned a question about this earlier, but we can actually be in an act of reciprocity and go into these areas and inoculate these trees with saprotrophic fungi that will then catalyze the process of decomposition and ultimately catalyze the process of soil succession in these areas. And it's increasing the biodiversity, and you can also grow local food and medicine in your local forest by culturing native species of fungi and adding them back into the environment. There's a couple techniques that I've listed down there about how to approach this. It's a huge topic, like I said, but this is just a little touch on it. Tissue culturing native species and then growing them on wooden dowels and actually physically inoculating stumps with these, which is seen in the first picture, which is another one of BAM's studies, the top picture. Inoculating fallen trees, making and distributing spore slurries of mycorrhizal fungi and saprotrophic fungi, tree plantings with native species inoculated with mycorrhizal fungi, so like taking the roots of seedlings and dipping them in mycorrhizal spore slurries, and native indigenous microorganism and mycorrhizal compost teas that you can add back into the soil. And this is really just, an act of reciprocity through positive ecological participation, which we absolutely have the ability to do. And that being said, just to leave us off, I'm just going to this last bit. An inextricably array of essential roles fungi play in the regulation and proliferation of global soil biology, hydrology, nutrient cycling, and plant, animal, and human communities are vital to incorporate in any eco-literate worldview and how we perceive ecological health, functioning, succession, and our role within it. So really, this is a huge way that we can be active in our ecological participation and how our role as humanity can add back into the systems for holistic health and resilience in human and non-human communities. Oops. Okay, so that is the end of that. And here's a lot more resources that I posted some on the homework page, but I can also share more if you guys are interested in having just more resources about all of this stuff. It's such a deep rabbit hole, all of these topics, but that's a good little overview of most of that.
Great, thank you so much,
Yeah.
Taylor. That was really exciting.
Excited to awesome. And I went a little bit over time. I do realize that, but if we want to, and whoever wants to stay, if we want to open up like a platform for questions for both I and Mia, yeah, feel free to shoot the questions. It's a lot of information, I know. Thanks for hanging in there, guys.
Can I go?
Thank you. Yeah, I just wanted to just acknowledge that if anybody needs to step off because it's after 5 PST, then please don't feel bad at all with checking out and leaving the meeting, but we're happy to stay on for a little bit longer so that there's time for questions. Go ahead.
I have 2 questions and they're for Taylor.
Yes.
How could I, with like test water for levels of contamination? And then the second question, how long did your burlap sacks, the ones that you did at your home, how long did it take them to myceliate?
Good questions. So there are a couple ways to approach soil and water testing. I bet, Mia, you could actually probably talk a lot more about that than I could. I know that there's some very simple tests that you can do just with like litmus tests, testing the pH of water and seeing how that's changing in the mycofiltration process, as well as teaming up with local universities and local labs to send in soil and water samples. That's really the ways that I've done it and approached it before. But even that being said, they're costly. They can be very, very costly depending on what you're analyzing for. So if you want to speak on that a little bit more, Mia, I feel like.
Sure, you know, it really depends on what it is that you're looking for. Like if you're worried about nitrate concentrations, you can extract nitrate with potassium chloride, do KCl extracts. But there's, yeah, it really depends on what it is that is your contaminant of question that you want to do some analysis on. But I agree that sending it to a lab using a mass spec or gas chromatography is a good approach.
Yeah.
I wanted to check like the before and after of like water or soil that was contaminated by like say an oil spill, and then test that like after and see how it compares.
Yeah, there's some specific tests from the EPA. I don't remember the number offhand, maybe like the 8160 or something for total recoverable petroleum hydrocarbons. But I used to use a lab in Petaluma called the Analytical Sciences, and they do a TPH, total petroleum hydrocarbons test for water. Or if you're really curious about a particular fraction, then you can look for that, because as the bigger, more complex molecule is broken down, you often see these smaller molecular counterparts.
Great, thank you.
And I think Daniela is going to cover some more of this as well. She's in an analytical chemistry lab at UCR. So she'll talk a bit about some of those analyses too.
Yeah. And then the second question, it was about a couple months that I let them grow out. Yeah, and based on environmental conditions, based on temperature and the different conditions and variables that I was talking about, you can definitely have different growth rates based on the just the aggressiveness of the species that you're using, as well as the temperature that they're growing at. It took about 2 months for ours to fully grow, but that can vary. Anyone else?
I have a question.
Yes, hi Patricia.
Did you actually ever find out if that filtration did take the E. coli or whatever out of the water system? Were you able to see if it worked?
Yeah, so unfortunately, there were no conclusive results from that study. We had, this was before my time, but just based on what I've heard about the study and what happened from it, there were no conclusive results because we were not able to get the funding or the ability to test the water and soil samples before and after. So it was really like an implementation strategy and not like a full conclusive study. But it's something that BAM does want to reapproach with a little bit more structure and to really go from start to finish and have that empirical evidence from soil and water samples. Yeah.
And then I was talking to an engineer, and he said that he knows of some chemical tests that can be done to see if there's aromatic carbons. Has anybody ever heard of any besides mass spectrometry? That's what we have tried to use here, but it's so expensive. Is there any kind of chemical test? Maybe Mia would know, or you know, to test for those aromatic carbons?
It's a little bit hard to hear you, but I don't know if you saw in the chat, there was a posting for Public Lab. And I know that some of the members of MycoRenewal have been consulting with public labs about different testing protocols that are a little bit more DIY. I'll just post that again in case people missed it, publiclab.org. Also, I just want to really underscore the importance of having controls. It's really tricky with a lot of these experiments because you could have an uninoculated control, but then there's lots of native or, you know, indigenous microbes that might be growing in the substrate that are doing a lot of work. So I think even having controls with inert substances that are not biologically, like not biodegradable, you know, like latex beads or something that is very inert, silica or latex, non-biodegradable substances as a control is another fantastic approach to then be able to compare, here's the inoculated treatment, here's the treatment that was not inoculated but is biodegradable, and then here's an inert material that's functioning just in essence as a physical filter.
I have one more just question. I kind of asked the same question of Willoughby. Is like we had a lot of success using worm compost to actually sterilize our substrate and also EM. And instead of sterilizing it, they actually worked together with the microbes to create way better mycelium than our boiled straw. So I was just wondering if anyone else you know of has been using microbes to not sterilize, but to like compete out the harmful bacteria. Because it worked really. I mean, sterilizing takes so long, so these soaks are quite short, like 10, 15-minute soaks, and then we would grow the mycelium right on the straw after it was soaked. I mean, we're in a southern climate; it's very hot and humid, so in like 8 days, so much mycelium, better than the boiled straw even. Has anyone else done anything like that? Do you know?
I know there's been a lot of like grassroots experimentation with practicing cultivation on non-sterilized substrates, but it's really hard to know because there can be beneficial or non-beneficial bacteria present in the substrate itself that can either compete or ally with the fungi. So when it comes to just like cultivation, it's always nice to use some type of substrate to give your targeted species of fungi a clean slate to grow on. But there have been studies done. I think I put one of the studies in the resources page, but I can check on that. But it's saying that, and it was a study done with empirical evidence saying that non-growing the fungi and using the filtration with non-sterilized substrate with the presence of both fungi and microbes actually had a better remediation ability than just the fungi or the microbes themselves. So there's that like innate synergistic capability going on there when there's presence of both fungi and microbes.
That's what we found. We were really excited. Like, did anyone else do this? Because we were trying to bioremediate incinerator ash, so we were trying to use both the microbes and the mushrooms together and see we had way more mycelial growth on those tests.
Yeah, you know, I kind of remember actually now, I did a remediation project after the Costco Busan oil spill that Paul Stamets was involved with and Lisa Gauthier from Matter of Trust. And I did all of the analyses afterwards, and we found that the compost from the Presidio mixed with the sawdust spawn and the wood chip spawn led to the greater reduction of petroleum hydrocarbons, much more than just the mycelium added alone. Paul was so excited with that result. He sent me this bound copy of Mycelium Running that was like this hardcover book, because it just like really underscored what we know about complex communities being more effective than just any one strain alone.
Yeah, that's why I was fascinated by the burned soil videos that we watched, how the mushrooms would come up, because one of the things that they found in Japan when they were doing research with microbes is that a lot of these beneficial microbes can survive in high heat. And because they couldn't sterilize them out of their beakers when they were doing experiments. So I also wonder, like, has anyone studied the relationship between the surviving microbes after the fire and the mushroom? Could there be a synergistic effect then as well? Because perhaps in trying to bioremediate, trying to create these biological communities with the mushrooms could really boost the degrading power of the mushrooms themselves.
Mm-hmm.
A lot of microbes found in compost are also pretty thermophilic. So yeah, that kind of makes sense.
Yeah.
Kristen has made a comment about being worried about mycotoxins from mold in unsterilized microfiltration bales. So yeah, that's a really interesting point. There's a lot of fungi that are naturally occurring, like Alternaria, for instance, Alternaria alternata, that produces mycotoxins that has a microbial antagonistic effect on other microbes. So maybe some of those would be good if you're trying to reduce pathogen loads, but if you're trying to do drinking water, it could be, you know, something to be concerned about. I am trying to ally with some researchers that do some sophisticated techniques to try to figure out what are these proteins and substances that are causing toxic effects on our lungs from lung exposure. But yeah, it's definitely a potent area of research.
Definitely a lot to be discovered there.
If there's time, I have a quick question for both of you, kind of about like life path and approach, because I'm still deciding, like, do I want to get a PhD like Mio, or would I rather just like be really on the ground doing community stuff? And I know that there's benefits to both, of course, and you're both doing academic and community work. So I'm just curious from your experience, like what's most frustrating about your work and like what's most rewarding?
So from my perspective, a lot of the reasons why I'm doing kind of the more on the ground grassroots community-based work right now is because it's just what's available for me, and it's definitely something that has piqued my interest for a long time. Like, how can we make these? I have a background in biology, and so I do have, you know, in understanding how academia can kind of like limit the accessibility of a lot of these approaches for people to be able to use. So I have a really deep passion for trying to figure out how to make these like complex topics more accessible for everyone to use, because like we're all players in this. And so that being said, I guess I it's not really like a frustration, but a limitation is being questioned a lot about the efficacy of these grassroots approaches, because they're not, you know, like created in the scientific method per se, which I totally understand based on my scientific background. I know that there's a lot of questions that can arise from not having, you know, like all of the elements of the scientific method laid out and not having the evidence from it. There's also a part of me that thinks that we can put a lot of trust in nature and nature's abilities to do what it's been doing for all of the past billions of years. And so it's kind of like, for me, it's both of those sides. It's wanting to like trust in nature's abilities to do what it does based on all of the research and evidence that we have, just putting it together in new and novel ways, as well as really wanting to, like my own personal path, I plan on going back to school, pretty much to do a lot what Mia is doing, of figuring out how to really ground bioremediation through scientific research in ways that can be like mass and widely produced, and just having more grounding in that. So I don't know if that answers your question, but
yeah, I'm happy to jump in too. So I focused on the grassroots approaches for quite a long time before I went back to grad school, and I was super excited about all of it. I had so much fun doing outreach and being really expansive to work with a lot of different communities that were affected by pollution or other disturbances. And I chose to go back because my insatiable appetite to learn about these processes just wasn't being quenched within my own access to materials. And I think things have changed now, that there's so much out there in online forums and publicly available resources that I don't think it's necessary to do that academic ivory tower approach. But for me, I really wanted to gather meaningful data to prove the efficacy of this work. And through you know, limitations with funding or the inability to like replicate things as much as I wanted to. I was not feeling like I was gathering the data that I was searching for. So I went on this journey with grad school, and I was even like a research technician for a while in between master's and PhD. And I found that some of the things that frustrated me at that phase was that a lot of times the money dictated what you put your energy into. So if I really wanted to focus on, let's say, polluted ecosystems, the lab that I was in, we weren't getting funding for that. So then I would work more in like in general degraded ecosystems. And then all of a sudden I'm finding that my identity is wrapped up in these stories that I was able to derive from research and collaborations that were the result of funding that I was able to get to obtain. One of the most exciting things about the field, or sort of the direction that I've taken, is that I've gotten to work on really large collaborations with some of my you know, favorite scientists, and that's been very exciting for me, like large scale collaborative research projects. And I feel like I can take that to really help build more tools in our tool set within this decentralized mycological research community to really take the research further and apply some of what I've learned in these more grassroots settings. But also what's been exciting is that while I have had to limit what I've been doing in order to get a dissertation written and publish projects that were initiated, the mycological community has been pushing this leading edge in so many impressive ways. And I come back to it and I just learn so much from so many different people. And it really is inspiring. And I just hope that some of the research that I'm doing can also be synergistic, and that we're just able to continue to push the edge and open doors for each other.
Yeah, thank you so much for sharing that. It seems like there's time in one's life to do many different approaches. So.
Yeah. For sure there is.
It feels like you can really do both, and there's as many people are doing the academic work, there's people doing the grassroots work, and we're all like working in synergy together. Totally. It's not like one or the other. It's really and both, like creating this field.
Right. And I guess I do just want to say that partially because the funding is a challenge, both at the grassroots level and at the academic level, there is something that is sort of exciting, which is that the great challenges and the most pressing issues of our time are the things that we're called to address. And then, you know, hopefully funding comes through from that. So while, you know, at one point it might be this particular degraded system, it changes. And for us to be able to be at that cutting edge of developing scientific tools or applying scientific methodologies in order to address these pressing environmental challenges, that's where it's at. All right. Well, with that, thank you. Yeah, with that, we should wrap up. It's we're over time, but this has been really fun to connect with you all. Yeah.
Thank you all so much.
Yeah, I look forward to seeing y'all next week.
Thank you
Thanks so
both,
much.
Taylor and Mia. Thank you.
Thank you. Yes, have a beautiful evening, everyone.
Right. Look in your inboxes for the next week's reading materials and homework. And thanks everyone for your great questions, and Mia and Taylor for putting together such detailed presentations and sharing your knowledge. Thank you.
Thanks so much.
Yes, thank you.
Bye.
Good night.
Bye-bye.
Bye.